Good day, everyone. Let me use this opportunity to welcome us all into today's class session. I am Mr. Oyedino Joshua by name, and I'll be taking us through the course ENA 203 titled General and Systemic Embryology. So, it is always good by the way of introduction that we know what the class session and the course we're about to take is all about. So this class session is divided into 30 weeks of teaching, which will be done via online Moodle teaching. So I would want to encourage us to endeavor to regularly check our Moodle profile and our portal to get information if there is need be. So without any further ado, let's delve into today's class session. So we'll be looking at the course contents first, which are spermatogenesis, oogenesis, ovarian follicles, ovulation, corpus luteum, menstruation, uterine cycle, hormonal control of uterine cycle, fertilization, cleavage and implantation, reproductive technologies, acronym which is IVF, surrogacy, embryo transfer. We also have the embryo manipulation and potency. We have the molecular embryology and transgenesis, gastrulation, notochord, neurulation. We have the derivatives of the germ layers, folding of the embryo fetal membranes, placenta, development of limbs, teratology, growth and perinatology, congenital malformations, which we'll be looking at the general introduction to that, cardiovascular development, and so let's look at the learning outcomes, in which within the course contents we are expected that at the end of this class we should be able to explain how the embryo forms from the zygote, discuss the role of cleavage and gastrulation in animal development, understand prenatal diagnostic methods and their significance, describe the structural features of primordia in tissues and organs, define risk periods in histogenesis and organogenesis, analyze the most common developmental anomalies. So, introduction. So, in this course, we are going to be exploring the fundamental aspect of human embryology from the formation of the gametes to development of organs and body systems. So here we'll be looking at the general embryology and systemic embryology. So the general embryology, which involves the early stages of human development, such as from how the eggs and the ova meet, and then how fertilization occur, and how this actually form what we call the zygote, and from zygote how this develop into a full-grown baby or into full-grown organs and body systems. So understanding these stages is for recognizing how various congenital anomalies arise and how prenatal diagnostics are utilized. So let's look at the formation of the embryo from the zygote. So if we are looking at the embryology, let's look from the pedigree, which is the origin of how all this started. So this is a diagram of how the cells, the egg cells and the sperm cells undergo the division. For anything to grow and for any growth to even occur, there has to be what we call the mitosis and the meiosis cell division, right? So the process begins with fertilization. After the sperm and the ovum form, that's when they produce what we call the zygote. So the zygote undergoes what we call the cleavage, which is a series of mitotic divisions producing smaller cells, which we call blastomeres. So eventually, a blastocyst is formed, which is composed of what we call the trophoblast, which is the outer layer and the inner cell mass. And this is what later becomes what we call the embryo. So let's look at the role of cleavage and gastrulation in animal development. So cleavage leads to the formation of the morula, which is also known as the solid ball of cells, and then the blastocyst, which implants in the uterine cells. So Actually is a series of rapid mitotic divisions of the zygotes. So which and despite the number of cells increasing, the overall size of the structure, funny enough, it doesn't change. So the zygote undergoes divisions to form a solid ball of cells called the morule, as I said earlier, and this division continues to form what we call a hollow structure called the blastocyst, which consists of what the inner cell mass, as I said earlier. So I told you earlier that this blastocyst then implant itself in the internal wall, and this process is called what we call the implantation. While the gastrulation is a crucial step that forms what the three germ layers, that is your skin. How do you think this is your skin that you are looking at? How do you think it comes to be? It's through this process we call what the gastrulation, and it has three layers. And if you look at this diagram, right at the right hand, we have what we call this is the ectoderm, the mesoderm, the endoderm, in which the ectoderm is the most inner layer of the germ layers, right? And we have the mesoderm which follows, and we also have the endoderm. So let's look at the prenatal diagnostic method. So when we as a baby, there are some tests or a way of knowing that, oh, there is something wrong with the fetus, or there's something wrong with this embryo, or there's something wrong with this baby while you are still inside the uterus. So we have many ways of diagnosing and observing the fetus till it is being till the baby is being born. So that what we call the ultrasound, which is safe, and it's a method that does not even involve you maybe penetrating or using an equipment inside the uterus to observe the baby. So it is a non-invasive imaging to observe the fetus, right? So we also have the amniocentesis and chronic villus sampling, which is CVS, also known as CVS, which is it is an invasive test to detect genetic abnormalities. So this is a test that is used to detect malformations, that is abnormalities during birth. So this has to involve in invading the fetus, right? So we also have the non-invasive prenatal testing, which also known as the NIPT. So this is a test that is usually done by collecting the blood, which is then analyzed to screen for any chromosomal abnormalities, and this is done without any risk to the fetus, right? So let's look at the assisted reproductive technologies, the molecular embryology, and what we call the let's look at the structural features of primordia in tissue and organ development. So primordia are just the early tissue structure that become what we call the organs. So for example, now we have what we call the limb primordia, which form the basis of arm and leg development. So these are what we call the humerus, the ulnar and the radius, these bones and the muscles like trachea and bicep trachea. So what actually develop, or what we can say to serve as the early tissue structure that now later become those bones, organs and muscles that you are seeing, is what we call the limb primordia. So we also have what we call the cardiac primordia, which later form the heart. So at different stages, the embryo develops these tissues precursors through interaction between the germ layers. That is, all these antecedent tissue, that is those tissues that are the early tissue structures. These are the ones that later develops and then interact together between the germ layers to form all these heart and organs that we are using now. So let's look at the risk period in histogenesis and organogenesis. So let us, what is histogenesis itself? Histogenesis is simply the formation of tissues, while organogenesis is the formation of organs, right? So during this formation of these tissues and organs, there are risk periods that malformation or abnormality usually occur. So you have from week 3 to week 8. This is a very high risk for abnormalities, to actually begin to develop. When a malformations usually begin. So during this period, you have to be very careful. And the first trimester, which is the exposure to teratogens, and these are harmful agents which can cause what we call the congenital abnormalities like heart defect or lane malformation. So this is a diagram of congenital malformations, that is birth defect, or when a baby is being given birth and at that particular moment is being born with some abnormalities. So let's look at the teratology and congenital malformation, which is the study of birth defect and their causes. So as I said earlier, congenital malformation is the structural or functional abnormalities which is present at birth. Examples which include neural tube defect, heart defect, cleft palate and limb abnormalities, and also which also include this club foot and spinal bifida. So mostly, let's look at the causes. What are the causes of these abnormalities that usually occur at birth? These are, we have the genetic factors, we have the environmental influences, also such as the teratogens such as alcohol or drugs. I also have the maternal health, that is the mother itself that's carrying the baby out. What is the health, or what is the level of the health of the mother? Is the mother itself okay? So these are many causes of the congenital malformations. So let's look at the specific organ development and electron micrograph. So here we are going to bring out each organ develop into what we see, into a full-grown organs now. So but then we won't really be going deeper since it's just an introduction to the course. So the cardiovascular system starts developing with the formation of what we call the heart tube, which then later folds and divides into chambers. So there are 4 chambers of heart. We have the left and right ventricle, and we also have the left and right, or what we call the atria. So let's look at the respiratory system, which begins with the lung bud emerging from the endoderm. So without wasting our time, let's also look at the electron micrograph, which this is what allows us to actually study the development of the organs in a high detail. That is, it helps to review the cellular architecture of organs as they form. So the cardiovascular system, this includes the formation of the heart and blood vessels, right, and also the partitioning of the heart into chambers. While we also look at the respiratory system. So while the digestive system, this involves the gut formation from the endoderm. So the division into the foregut, midgut, and hindgut, forming organs like the liver, stomach, and intestine. This is how the digestive system comes to being. So let's also look at the urinary and genital systems, which is the differentiation of the mesonephric duct into kidneys and reproductive organs, and also the early urinary structures like the pronephros, mesonephros, and metanephros, which develop into what we now call the adult kidneys. So in conclusion, embryology, you agree with me that embryology reveals the incredible journey from a zygote to a fully developed human. So by understanding this process, this helps us to aid and enhance our ability to detect and address any potential issues at least at an early stage through prenatal diagnostics and all these tests that we mentioned of earlier. So I will also recommend that advancements in reproductive technologies which offer hope for many families, while ongoing research ensures that we continue to improve what we call the maternal and fetal health. So please, if you have any question, let's drop it as a comment section. So I would like to remind us again that we should regularly check our Moodle profile and our portal to get information. So see you next class. Thank you very much for listening.